Welcome to the short course in the microbiome. This is a free series that will run through April. We have a bonus session in May, and then we also have a gathering in Fort Detrick in September that you are hereby invited to attend. I'm just going to tell you a little bit about our organization. I'm Joanne Geer. I'm the Executive Director of the Biopharma Research Council. And essentially, our mission is to bring scientists together across the many silos of biomedical research. So that's industry, academia, nonprofit, government, and supplier labs, and those who love them. And we reach out, as you can see, in a number of different ways. That's me. I really love this slide. This is from a conference that we ran last summer called D2D, Data to Drugs and Diagnostics. I feel it really expresses, that's Bob Boland from Janssen Pharmaceutical giving that talk. I'm just going to share with you a few of the things that are coming up in April. Also this month, Pardis Sabeti is going to be speaking as part of, we have an occasional series, so it's not a short course, it's just a series on global collaboration to address Ebola, and we're very excited to have Pardis Sabeti speaking for us. She's been profiled in the New Yorker and the New York Times and just has an amazing breadth of experience and connections and insight. So it's going to be a beautiful program. In May, we're going to be in Boston. This is a symposium that will look at CRISPR and zinc fingers and talons and such, the technologies, the models, and how some companies are starting to actually move toward business with these new technologies. This is hosted by Merck. They very much want people from other companies to attend because they are looking for collaborators. So we're reaching out to other pharmaceutical companies and biotechs as well as to academic institutes and those other groups that I identified earlier. In October, we go back to Research Triangle Park in North Carolina, and this is a really nice one-day program, and we have a committee working on it. We think the theme this year is going to be on technologies that are relevant in human biology, animal biology, and plant biology. Just going to share with you, I'm going to go quickly, I'm not going to name everyone, but we have a really interesting group of young investigators who are involved in various aspects of biomedical research. We have a terrific scientific advisory board. Profiles of all these people are on our website. But I want to share this with you because if you are intrigued to get involved, you are invited to join this illustrious group. These are some of the awards that we gave just recently for activities in 2014. And I'm just sharing that we're connected with a number of other organizations that touch on different aspects of the biomedical research community. And this has really helped us to grow our impact. We have a group of about 8,000 researchers that we communicate with regularly. This is just a wide range of organizations that have helped us just in the past couple of years, and it's not even everyone, but we've been very blessed with great interaction, collaboration, and partnership. And now we come to those organizations that have really made today's program possible. Intech is an open source scientific publishing company and has done really terrific work. IES Diagnostics is where Winston Kuo, who you're going to meet in a moment, comes from. The NIH is where Howard Young comes from. And Planet Connect is essentially the principal founding sponsor of the Biopharma Research Council. They do conferences, internal conferences for big pharma and biotech companies so that when major companies want to gather all of their researchers together, Planet Connect organizes that. But of course, those are all secret meetings, so we can't say anything more than that. Please feel free to enter a question in the box at any time. We're going to hear from our primary speaker, Rob Knight, first, and then we're going to open for discussion with the other two folks I'm going to introduce to you. And then please enter any question you have in the box, and we'll take a look at those and try to address as many as possible. Here we are. Welcome. I'm just sharing this. This webinar happened last December, and this was a nice introduction to thinking about the microbiome that led to the series we're in today. And the recording of this is on our website, so I just wanted to show you that today. Okay, so I'm going to introduce to you our speakers. First, we're going to hear from Rob Knight, and then Winston Kuo and Howard Young will be joining the conversation. Rob Knight is a professor of pediatrics and computer science and engineering and director of the Microbiome Initiative at UC San Diego. He is the co-founder of the Earth Microbiome Project and the American Gut Project, and co-founder and chief science officer at Biota. Dr. Knight has co-authored more than 300 peer-reviewed research articles and has been cited more than 41,000 times with an H-index of 86, according to Google Scholar. And that was a few months ago, so maybe it's even better than that. He received his PhD from Princeton in 2001 and was at the University of Colorado Boulder and from 2001 until just recently. Dr. Winston Kuo is the COO of IES Diagnostics. He is the founder of the Howard Medical School's Laboratory for Innovative Translational Technologies. He sits on the editorial boards of multiple peer-reviewed journals and is editor-in-chief of the Journal of Circulating Biomarkers and Nanobiomedicine, both of which he founded. He has consulted with the NIH on interdisciplinary research and sits on a number of business and nonprofit scientific advisory boards, including the Biopharma Research Council. He received his Doctor of Medical Sciences, Oral and Computational Biology from Harvard Medical School. And finally, Dr. Howard Young received his PhD from the Department of Microbiology and Immunology at the University of Washington. Joined the National Cancer Institute in 1983, becoming Deputy Chief of the NCI Laboratory of Experimental Immunology in 2006. Most recently, his lab has developed novel mouse models of lupus and aplastic anemia. He is also collaborating with a laboratory in Malaysia on the development of probiotic microorganisms as carriers for vaccine delivery systems. So thank you. So we're going to turn it over to Dr. Knight.
Great. Thanks, Joanne. Let me just get the slides up. And can you see that okay?
Yes, looks good.
Great. So, okay, so I thought I'd talk a little about the systems approach, both in the sense of systems biology and in the sense of ecosystems biology, which is really what we need to use to understand these very complex microbial assemblages in our gut and elsewhere in our body. There we go. So, so humans have always asked a lot of interesting questions about life, and this dates back to things like the Lascaux cave paintings 30,000 years ago. So you can see that people have been interested in things like what kinds of animals are there, or as we'd call it today, taxonomy. How many kinds of animals can I find in different locations here in the context of hunting? So in technical terms, we'd call that alpha diversity. How different is one place from another, which we call beta diversity, the dissimilarity in communities in different places. And you can also see that there's a fair number of trophic interactions going on there with finding out which organisms are eating other organisms, which is also very important in the kinds of in the kinds of research we're talking about. So how do we describe and compare diversity? Well, ecologists have been struggling with this problem for between decades and thousands of years, depending on who you define as an ecologist. But one of the key concepts is these different kinds of diversity. So alpha diversity is really trying to figure out how many species or how many kinds of organisms are in a particular sample. So in this case, if the colored circles represent community A and community B, you can see that in each of those cases they have the same number of colors, so you might say that the communities have the same alpha diversity. And so this is useful for things like trying to figure out are polluted environments less diverse than pristine environments. In the context of disease, disorders like obesity have been linked to low alpha diversity, so a degraded ecosystem in the gut. Then with beta diversity, what we're concerned with is how many species are shared between different samples. So in this case, there are two shared colors between community A and B, the red one and the dark pink one, which you can see are present in both of those communities. And so the kinds of things we're asking with beta diversity are things like, does the microbiota differ between different disease states? So in other words, can you tell that there are particular kinds of organisms that differ between lean and obese people? Another important distinction is qualitative versus quantitative measures. So with qualitative measures, we're just looking at the presence or absence of particular taxa. For example, for alpha diversity, you're talking about how many species are in a particular sample. For beta diversity, you're talking about how many species are shared among samples. Whereas with quantitative measures of diversity, we also consider the relative abundance. So we're concerned about evenness, for example. And for example, in alpha diversity, population A and population B have the same number of colors, but you can see that in population A there's a lot of red ones, and in population B they're more evenly distributed. So we would say that B was more diverse than A on that measure of alpha diversity. Then in beta diversity, what you're primarily concerned about is whether you care about only similarities in the common species, or whether you also care about similarities and differences in the rare species. And then another thing that's really important in measuring the diversity of an ecosystem is how you measure that diversity in terms of the phylogenetic tree, or if you're just doing a taxonomic inventory, kind of like you would use for a gene list if you were doing a microarray study. So with taxonomic measures, we care about how many species are shared between samples, whereas with phylogenetic measures, we care about how much phylogenetic distance is shared between samples. So in a taxonomic measure, community A and community B would seem the same because they each have 6 distinct colors. But then if we put those colors on a phylogenetic tree, and the more similar colors are more closely related, in that case, we would say that the organisms in A were less diverse because they cover a lot less of the phylogenetic tree than the organisms in B. And the reason why we care about this is that phylogenetically related organisms are more likely to play similar roles, just because they're evolutionarily related. And when you use taxon-based methods rather than phylogenetic methods to measure biological diversity, it's not that you assume no phylogenetic tree. You assume a very specific star phylogeny where all species are considered equally related, and throwing away that information that you have about the phylogenetic tree that relates different species can really be a mistake, as I'll show you later on. So one key challenge in ecology is how do we apply these principles, which have mostly been worked out in the context of large animals that you can see around you as you look around the planet. How do we apply these principles to microbial ecosystems and different contexts on our planet? So the way we do this is we use the ribosomal RNA, which is an excellent phylogenetic marker for several reasons. It's found in all species. It has slow-evolving regions that let us construct PCR primers, and then fast-evolving regions that allow us to tell the differences between even relatively closely related species. And then there are large pre-existing databases for comparison with the sequences from known species. So we can reconstruct relationships at different depths, and then map them all onto one evolutionary tree that we can use to compare different communities. And in this context, it's important to remember that our world is very much a microbial world. So this is a tree of life put together by Norm Pace's lab a while ago now, and I've taken the liberty of highlighting all the multicellular lineages in red on this tree. So everything that's in black is microbial, including all the archaea, all the bacteria, and most of the eukaryotes. And on the scale, this is us, for example, and this is corn. So if you had cornflakes for breakfast, that's how much of the diversity of the tree you were covering. This is a mushroom, and all of the rest of this is microbial. And the vast majority of these microbes that contain most of the diversity, we don't yet know how to culture them. All we know about them is the DNA sequences that we use to build these kinds of trees. So you might think that what we need is a field guide, or as it's often called in systems biology, a parts list, kind of like National Geographic has like a field guide to the birds of North America that tell you what they look like, where they are, and so on. But for microbes, we might need a few more pages to do that, because rather than 990 species of birds, what we're talking about is an estimated 990 million species. So there's this tremendous diversity in the microbial world that we're only just beginning to understand. And so how we find out about that diversity is by a technique called direct environmental sequencing that lets us see the other 99% of microbes that we don't know how to culture. So the principle behind this is that we get samples and extract the DNA. So maybe the red sample comes from a patient, the blue sample comes from a healthy control. And most of what I'm going to tell you about, we PCR amplify the small subunit ribosomal RNA gene and use that as a marker, like I showed you for the different organisms in an environment. But you can also do shotgun metagenomics, looking at all the DNA in the community, and other molecular techniques that I'll talk about later. So then traditionally, you would clone and sequence, but the great thing about modern sequencing techniques is you don't need to clone anymore. So every time you run the HiSeq instrument, you get a few 100 million sequences. And so you're saving yourself a few 100 million toothpicks, a few 100 million cloning files, and frankly a fair amount of time and effort by sequencing from the PCR product directly. Then traditionally what you do is you blast those sequences against GenBank and see what they were similar to. But ironically, the success of direct environmental sequencing makes that not so useful anymore, because you're not going to hit E. coli or B. subtilis or some other organism where you know what it does. You're going to hit uncultured environmental sequence number 93,000,007 from one of these studies. So instead what we have to do is we have to take an explicitly evolutionarily and phylogenetic approach where we build multiple sequence alignments, we build phylogenetic trees, then we relate the tips of those trees, so the modern sequences that we observed, back to the communities that they came from. So both marker gene approaches like I'm showing you here and shotgun metagenomic studies where you look at fragments of all the genes can be thought of as producing abundance tables. And so what we're doing is we're taking DNA from fecal samples from all over the world. And then with small subunit ribosomal RNA with targeted gene sequencing, for each taxon, for each sample, we get the frequency of that taxon in that sample. Then for shotgun metagenomic sequencing, we have more or less the same thing, except for each sample, we have the abundance of each gene in that sample. And you can use these kinds of data for a whole suite of downstream analyses, ranging from alpha diversity, where you can ask what genes or most functions are most diverse in particular taxa. For beta diversity, you can ask whether groups of samples separate either in terms of their microbes or in terms of their functional genes. You can do machine learning to try to discriminate the samples from one another. You can build co-occurrence networks either at the taxon level or at the gene family level to try to understand which microbes and which genes are working together. And then finally, you can do things like metabolic network modeling, where from the gene functions, either directly observed or inferred from the microbial communities, you can start to build systems-level models and do things like flux balance analysis to start to understand what the ecosystem would do at steady state. Of course, it's important to recognize that there's a lot of other omics out there, and so this is something we did a few years ago in GigaScience, just counting the number of words that ended in omics as a function of time. And so you can see that as of 2009, there were articles with over 200 different omics words. But it looks like we may have reached peak omics in 2009, and perhaps the number of new omics terms that are proliferating is finally starting to decrease. But there's only a few main kinds of omics that you need to worry about in the context of microbial analysis. So of course there's genomics, looking at whole genomes. There's metagenomics, where we're looking at fragments of DNA. Metatranscriptomics, where we're looking at fragments of RNA. Metaproteomics, where we're looking at fragments of proteins. And then finally metabolomics, where we're looking at the frequency of small molecules. And all of these produce essentially the same kinds of tables, where this lists of the objects that we're interested in, and then there's the frequency of each of those objects measured across a whole set of different samples. And so many of the computational and other analytical tools downstream from this are exactly the same, no matter which kind of table you're looking at. So to handle this kind of complex multivariate data, we built this pipeline called QIIME, initially developed by Greg Caporeso, Justin Kaczynski, and Jess Dunbar when they were in my lab, and now developed by a wide range of researchers worldwide. So it stands for Quantitative Insights into Microbial Ecology. It's free, it's open source, it runs anywhere from your laptop to the Amazon EC2 cloud to the Hopper supercomputer at NERSC with 140,000 cores, which is the largest system we've run it on so far. And so what we do in the lab is we take 16S ribosomal RNA genes from hundreds of different samples using DNA barcodes. We mix them together. We used to sequence them on the 454 instrument, but subsequently we've moved to the Illumina platform, which is both more accurate and gives us a lot more sequences. Then because each sequence has a barcode, we can tell where it came from. We clip off the barcodes, build alignments, build trees, and then use the trees to cluster the samples. So we might expect the healthy versus the disease samples to separate. But we do a lot of other analyses downstream as well, everything from classical statistics to machine learning. And so a lot of what QIIME does is it resolves problems with noisy next-gen sequencing data, including different kinds of pre-processing, things like denoising and other kinds of quality filtering, grouping the sequences together into what are called operational taxonomic units, where you can find out essentially what the species level taxa are, doing alignments and trees, and then building and then doing things like taking the OTU table, so the per sample counts of each taxon, doing alpha and beta diversity measures, and then doing a lot of different kinds of data visualization tasks. By the way, I should mention that the choices made in the pipeline really matter a lot. So I'm not going to go into too much of the details of how Unifrag developed with Kathy Lazapone when she was in my lab. It's one of my first grad students. I'm not going to go into too much detail about how this distance metric works, but essentially what it does is it uses a phylogenetic tree as a measuring stick to compare two communities in terms of what's unique in evolutionary history in each of those communities with respect to the other. And so when we do clustering of environments from around the world, we see a very clear split between saline communities in green and non-saline communities in blue, with estuaries which are truly intermediate because they mix saline and non-saline in the middle there. But when we use taxon-based measures that throw away the phylogenetic tree and just look at counts of each species or of each genus as though they were all independent, you can see that with things like Kozinski quantitative symmetric measure and Euclidean distance, you get these spike artifacts at 90 degrees relative to one another. And the reason for that is that having the tree allows you to better reconstruct the evolutionary history and get a better picture of what's going on. I should point out that it matters a lot which region of the 16S ribosomal RNA you choose, and it can have large effects on clustering. So this is work that Zhongji Liu did in my lab a while ago when he was a postdoc, just taking near full-length sequences and clipping out little pieces of them to ask, with a short read like you get from next-gen technologies, do you read out the same community? And to cut a long story short, the answer is basically yes. So what we're looking at here is a clustering of some human samples, some samples from mice, and some samples from the Guerrero Negro microbial mat, a hypersaline mat that's among the most diverse microbial communities that we know of on Earth. And if you pick a good region of the sequence, like going forward from position 517 relative to the E. coli sequence, you get exactly the same clusters right down to the same outliers. Whereas in contrast, if you pick a bad region of the sequence, you make the amazing discovery that some of the humans look more similar to mice than they do to the other humans. So at first this would be very exciting, but then you'd be sad later on after you went back to your clinical collaborators and asked, what is it about these people that makes them resemble the mice? Is it that they're vegetarian or very tiny or live in a restricted environment or what? And then when you got the full-length sequences and found out that it just wasn't true, you'd be disappointed. So it is important to pick the good regions. But the good news is that although the 16S ribosomal RNA is still evolving, it doesn't evolve very quickly. And the same regions that were good a million years ago or that were good 10 years ago when we did the initial characterization are still the regions you should use now. I should also point out that how you do taxonomy assignment can matter tremendously. So again, when we just clip out parts of a sequence and see if the taxonomy assignment is the same, what we found when we did this back in 2008 is actually the results depend more on what taxonomic assignment method you use and what region of the sequence you pick than whether it comes from a mouse or from a person or even from a hypersaline microbial mat. So it's really important to be consistent in your methodology, both in the lab and in your bioinformatics if you want good results. So one thing you might be wondering is how plastic is the microbiome, given that there's a tremendous amount of diversity in adults. And this is data from the Human Microbiome Project main papers, which we published back in 2012. And what you can see is that even in one body site, like for example the gut, these are at the genus level, and so some people consist of essentially 90% Bacteroides in the gut, whereas other people only have about 10% Bacteroides, so that's this taxon in red. And for many of these other taxa, you see that there's tremendous variation at the genus level. When you look at things like clinical states like obesity, what we're looking at here is some data from a paper we did with Jeff Godden's lab back in 2009, led by Pete Tenbaugh, who's now a faculty member at UCSF. And what you can see is, again, this tremendous amount of variability in the, for example, the firmicutes in red versus the Bacteroidetes in blue, one person to another. There's even a fair amount of variation just one time point to the next when we sample the same person twice, although the same subject is more similar to themselves on average than they are to other people. However, even from this noisy data, we can tease out very clear patterns. So in this case, for example, Dan Knights, who was in my lab at the time and is now a faculty member at the University of Minnesota, was able to take this noisy data, where you can't tell who's lean or who's obese just by looking at it, and show that when we ran it through a random forest classifier, we could actually tell who was lean and who was obese with 90% accuracy based on this microbiome data. So on the one hand, this is kind of cool that you can tell if someone's lean or obese with 90% accuracy based on their microbiome. On the other hand, we don't think it has a lot of commercial potential for those of you listening from companies out there. We don't think it has a lot of commercial potential as a test for obesity, because there's probably easier ways to tell if you're fat than sequencing all your microbes. But on the other hand, if we try to do the same classification task based on human DNA, you can only classify someone as lean or obese with about 60% accuracy based on their human genes versus about 90% accuracy based on their microbial genes. So one thing you might be wondering, because a lot of microbiome work is done using fecal samples, is whether fecal samples are actually okay for representing the gut. And so this is a reanalysis we did of data from Dave Relman's lab, where they looked at 6 sites along the distal large intestine, plus matched stool samples from 3 different subjects. And so when we analyze the data with these methods, when I color by subject, you see all the samples from the first person in red, and the second person in yellow, and the third person in blue on the left-hand panel, all cluster together very nicely. Whereas if I take the same data and color it by body site, you can see that the stool basically clusters with the rest of the samples, although sometimes there's a little bit of variation, but it's small compared to the difference between different people. So the great thing about being able to use stool samples is it really helps us put our human microbiome in context. And so this is work we did with Ruth Lay, then in Jeff Godden's lab, now at Cornell University, looking across a whole range of different mammalian species, first at the primates, and then more broadly within the mammals. And the great thing about being able to use fecal samples for this is, as Ruth was essentially able to go to the zoo, specifically the St. Louis and San Diego zoos, and work from fecal specimens, which is great because it's really hard to do a colonoscopy on a bear or a rhinoceros. So when we analyze these with the unifrac technique and principal coordinates analysis, like I was showing you before, what we see is very nice clusters based on diet and gut type and lineage. So you can see the herbivores in green separate from the omnivores, in blue separate from the carnivores in red. But I wouldn't feel good about telling you about a systems approach if I didn't show you some incomprehensible network diagrams. So I'm going to show you some of those. And what this is a bipartite graph, where what we did is we had one node type for the fecal specimens, then a second node type for the operational taxonomic units, so the kinds of microbes. Then we connected those points together with an edge that was weighted by how often we found that kind of microbe in that fecal sample. And so the fecal samples are colored by color, are colored by diet types, so the herbivores in green, the carnivores in red, and the omnivores in blue. But actually this network diagram, unlike many hairballs you've seen, is not actually that incomprehensible, because again you can see one very nice cluster for the carnivores, like you can see in the PCOA, where that includes everything from bears to cats to hedgehogs and echidna and so forth. You see 2 clusters for the herbivores, and fascinatingly what this is the difference between hindgut and foregut fermentation. So 2 different ways of fermenting grass, essentially, that have evolved multiple times in the animal kingdom. And then we see one group of omnivores, including everything from rodents to bats to primates. And if you're wondering where we fit in, basically we as omnivorous primates just fall into the rest of the distribution, despite the diverse habitats and the diverse diets that humans have explored. We can also use this sort of thing to look at development. So, for example, in additional work with Ruth Lee, we can understand how the microbiome develops in infants, starting with the meconium, and then going all the way over the first 2.5 years of life to resembling the mother in terms of the stool. And then you can see that this is coupled with a relatively steady increase in diversity over those first 2.5 years of life. So you get what looks like a relatively clear and simple pattern, although we'll come back to that a little later. And because the effect of development is so large, we can even do this cross-culturally. So here I'm showing you what we did with Jeff Gordon and a host of other collaborators, looking at kids from ages 0 to 18 on the x-axis. Then on the y-axis, we're looking at the average difference between each child's sample, the average of the adults for the same community. And you can see that for an African sample in red, for a South American sample in green, and for the US sample in blue, the approach to the adult state is very consistent and happens over the first 3 years of life. However, where you wind up is totally different. So here's principal coordinates plots of the same data. And what you can see is that age is the main effect, going from the youngest kids to the oldest adults, like you'd expect from the data I'm showing you in the main graph. But the final populations, the Western population in blue, is totally different from the two non-Western populations, although they separate from one another along another axis that goes into the plane of the screen. And in this context, it's important to remember that even large-scale projects like the Human Microbiome Project and like MetaHit are only looking at healthy Western adults. So they're only looking at this very small fraction of the space of possible microbial community configurations. And as soon as you look at kids, and as soon as you look at non-Western populations, you see these totally different configurations of the microbiome that you just miss if you're looking at healthy Westerners. In a drug discovery context, this is really important as well, because we now know that the microbiome affects drug responses from everything from acetaminophen to cyclophosphamide. And so if you're going to test your drug on one population, say where the drug trials are a lot cheaper, and then deploy in another population where the microbiome and potentially the pharmacokinetics and the toxicity are completely different, that's certainly taking a bit of a risk. So one thing that's fascinating is that totally different species assemblages can lead to very consistent functions. So this is from that Tenbow et al. 2009 paper I showed you before, looking at the lean and obese twins. If you look person to person, even at the phylum level, this is with shotgun metagenomic data, not with 16S ribosomal RNA. What we see is tremendous variation in the amounts of even dominant taxa, like the firmicutes in light blue versus the Bacteroidetes in dark blue versus the actinobacteria in whatever that color is, peach, I guess. But when we look at the functions obtained from the same data, you can see that the functional categories are extremely consistent, one sample to the next and one person to the next, whether they're lean or obese. And we can't explain this just in terms of the gene contents in the genomes. So you might think that on average, Bacteroidetes is going to be pretty different from a firmicute, but it turns out that although that's true, the difference in those major taxa would lead you by chance to expect much more difference between the different samples, which remember are taxonomically different from one another than we actually see. So it looks like there's some kind of selection to keep everyone's functions relatively consistent, even when their taxa vary tremendously. And we saw this again in the Human Microbiome Project. So here's what I showed you before, looking at phylotypes in the gut. Here's the matched data, looking at the pathway level, and you can see that whether we're talking about the gut, or the skin, or the vagina, or the mouth, you see the same pattern of very consistent molecular functions, but totally different species assemblages leading to those functions. So one thing that's really exciting is that we can match data up across these different levels. For example, matching up 16S ribosomal RNA with metagenomic data using a technique called Procrustes analysis, which we introduced in this paper led by Brian Merge in Jeff Gordon's lab back in 2011 in Science. And essentially what we're doing here is we're taking different point clouds and then we're trying to superimpose them on one another by rotating them, stretching them, scaling them, and so on. So if you have the blue and the red point clouds, then you can line them up just by rotating them around. Whereas, for example, if you have the blue and the green point cloud, if you line up the A's with each other, you can't get the B's and C's to align, for example. So what we can do here is we can see that the taxonomic data with 16S matches up very nicely with gene functions, whether we're looking at KEGG orthology or carbohydrate active enzymes in CASE, or proteases and merops, or EC numbers as a measure of enzymatic function. So we get this very good relationship at these different levels, going from taxonomy to function. Let's see. Yes, actually, yeah. So anyway, so you can match up these different levels to one another. Now, as I mentioned before, the HMP only covers the start of human diversity, so we really need to look at different ages, different populations, and people with different health status. And as I mentioned before, we see these profound changes with population, looking at the Western populations versus non-Western populations. And this is what you cover when you're looking at the HMP or looking at MetaHit, which is the European equivalent project. So although these projects are very useful for defining a baseline within the healthy adult Western population, there's all these other populations you might want to see. So this led to this challenge in integrating the HMP with the universe of other data that's out there. And this effort was largely led by Antonio Gonzalez in my lab, where the idea is that what we can do is we can take individual data sets where we use QIIME to upload and verify your sequence data and then your metadata that's compliant with the Genomic Standards Consortium standard called MEMARCS. Then we use a technique called reference picking to get OTUs into one reference alignment. Then you can download your table and the HMP OTU reference tables and mapping files. Then what we can do is use StarCluster to create an instant QIIME cluster on Amazon Web Services. So essentially what you do is give Amazon your credit card number, and frankly I'm guessing they already have your credit card number, right? So essentially what happens is you just create this instant cluster, run your analysis for a few dollars, and then let it dissipate. So what you do is you merge your OTU tables and mapping files with the reference ones, and then you can verify, filter, do analyses in QIIME, and then repeat that until you have a result that looks nice. So one thing we can see here is that the overall cluster patterns in the Human Microbiome Project, that's shown here, match very nicely with the cluster patterns that you get when you look at all the data that was available prior to the HMP. And so the gut's colored in black, the oral samples colored in green, and so forth. And you can see that it matches up very nicely when we overlay them. Although for some things like the stool, for example, we can see differences where, for example, the HMP did not cover infants. So we see these kinds of community configuration that we just didn't see in adults. I'll just show you one really compelling example of why you want to do this kind of data combination effort. And so the infant time series I showed you before, this is what happens when you combine it with the HMP data. So what we're looking at is we're looking at an animation put together by Antonio and by Yoshiki Vasquez, a grad student in my lab, where we're starting with the infant fecal community, and you can see that it actually looks more like a vaginal community. We'd actually expect this based on this kid's delivery mode, because you get coated with the vaginal microbes and during the delivery process, and they even get into the meconium. So what we're going to see is an animation of that infant time series I showed you before that looked like that smooth progression to the adult state, shown on the data frame of the Human Microbiome Project. And so I'll start this going. What you can see is sometimes 1 week to the next there's tremendous change to the microbiome. Other weeks it's relatively static. And remember that what matters on these diagrams is the distance between two successive points. So what we're seeing is that 1 week to the next, the change in this kid's gut microbiota can be larger than the difference between any two healthy adults we saw in the HMP. So coming up here is something pretty cool. So the kid gets antibiotics for an ear infection. You see this tremendous regression of the microbiome followed by recovery. So that went by pretty fast. I'll just rewind that and play it again for you. What you see is that the kid gets oral antibiotics for an ear infection. You see this regression of the microbiome undoing months of normal development, followed by a relatively rapid recovery. And then by the time he gets up to 2.5 years, you can see he's more or less in the healthy adult state. And this is just fascinating because you couldn't see these patterns at all when I showed you the data set alone. We can only understand them when we combine them with these large-scale resources like the HMP that NIH has put together. Okay, so a major thing you might be wondering at this point is how can I tell cause from effect? And so there's this classical idea of Koch's postulates, where the idea is that you have to find the microbe in all organisms that are suffering from the disease, but not in healthy microbes. You have to be able to isolate it in pure grow it in pure culture, then show that it causes disease. And then you have to re-isolate the disease, re-isolate the organism from the individuals that you infected. But of course, this is really difficult to do in humans for ethical reasons, and additionally, the large majority of the microbes that are out there, we don't necessarily know how to grow them or to isolate them, so it's really difficult to meet these kinds of criteria. So we can do a lot more of this stuff in mice, and what I'm showing you here is the gnotobiotic isolation units in Jeff Godden's lab at WashU, where essentially what we can do is we can grow the mice germ-free in a bubble without any microbes, and then infect them with the microbes that we think are important. So what's great about this is that we can start to take that very complex microbiota and then decompose it into a parts list of individual microbes, and then look at different combinations of those microbes and ask how much of a difference they make when you leave them out or when you put them in. So you might wonder if these differences actually matter. Obesity was one of the first things we studied. So this was a paper led by Ruth Lay back in 2005 in Jeff Godden's lab. And so what we're looking at is normal mice versus leptin mutant, which is about 3 times the mass of a normal mouse. And you can see that it also has a profoundly altered microbiome with more firmicutes, fewer Bacteroidetes than its littermates who are lean and are either heterozygous or wild type. So then with Andrew Gewitz's group then at Emory, we looked at TLR5 knockout mice, which also get fat, but for a different reason. So here are the TLR5 knockouts. You can see they're very fat compared to a wild type, with larger fat pads in both the males and females, higher triglycerides and cholesterol in the serum, and even high blood pressure. And again, you can see that the TLR5 knockouts have a profoundly altered gut microbial community from the wild type mice. So what's different? What's interesting is the difference in mechanisms. So with the OB mice, other researchers in Jeff's lab basically showed that you could transplant some of the obesity phenotype to a completely genetically normal mouse that was raised germ-free by transmitting the microbes. And what was going on there was an energy harvest phenotype. So if you did bomb calorimetry on the fecal pellets, there was just less energy left behind, presumably because the gut bacteria are more efficient at extracting it. But with the TLR5 knockouts, what's going on is very different. What happens with the TLR5 knockouts is actually they're more efficient at extracting the TLR5 knockouts are no more efficient at extracting nutrients from the diet. So when you do bomb calorimetry, you see no difference in the fecal pellets. Instead, what happens is they actually eat more, which is why they become obese. So it's essentially all a behavioral phenotype. Again, it's a behavioral phenotype that you can transmit to a wild-type mouse that's been raised germ-free. You can cure it with antibiotics, and you can also cure it by just putting in its cage the amount of food a normal mouse would eat. So we have no idea how this works, but Andrew did have this cartoon drawn in lieu of an actual explanation, just to remind you that there's far more microbes in your body than there are your own cells. They may be outvoting you when it comes time to make choices at the cafeteria. You might wonder if any of this extends to humans, and there's some very intriguing evidence that suggests that it does. So again, this is some work from Jeff's lab that I wasn't involved in, looking at people on a diet for a year, where the initially obese people came to resemble lean people in terms of their microbial community. Then the more weight they lost, the bigger the change in abundance of particular kinds of microbes, whether they were on a carb-restricted or a fat-restricted diet. And as I mentioned, we can classify people as lean or obese with 90% accuracy based on their microbiome. So where we're going with this is we're trying to develop a kind of personalized medicine based not on subtle differences in the human genome, but based on obvious differences in the human microbiome that we can deploy even in developing nations. This photo was taken by Tanya, one of Jeff's grad students at the field site in Malawi, at the malnutrition clinic that we work with there. And although food is scarce in that population, you'll notice that, for example, they do have cell phones. And the remarkable fact is that there are over 8 billion active cell phones on Earth now, which is pretty impressive when you consider there's only 7 billion people. Of the billion poorest people in the world, about 20% of them have their own cell phone. So it's a technology that's just penetrated everywhere, in large part because the digital signal processing that you need to set up a cell network is so cheap that it's effectively free. And this is true not just in Malawi, but pretty much everywhere. So I took this photo in Bangladesh when I was there in 2012, teaching a cloud computing workshop at ICDDRB. So you can buy a sizable hunk of a cow in relatively bad parts of Dhaka. And then right next to that, you can have your cell minutes recharged. So anyway, as I mentioned, the reason for this is that computing is getting cheap. But one thing that's getting cheaper even faster than computing, which is represented by this white line, is DNA sequencing, which is represented by this blue line. So in the time that computing got 100 times cheaper, so this is on a log scale, DNA sequencing got almost 1 million times cheaper. And so the idea is that the germ-free mice are very expensive, but if we can move it to an in vitro assay, or ultimately a DNA sequencing-based assay that's so cheap it's effectively free, we might be able to predict what happens for particular people with different therapeutic interventions. So what we're doing is we're doing these studies in humanized mice in our, this is with Jeff Golden's lab at WashU. So in our NIH-funded project on obesity and our Gates-funded project on malnutrition, the idea is to take the microbes from people, put them into germ-free mice with no microbes of their own, then see whether the mice gain weight or lose weight. And so the data from this are just beautiful. So here's time on the x-axis. Each interval is about 2 weeks versus principal component 1 of the microbial community. 5 different mice colonized with the same donor. You start them on mouse chow, then you switch them to the Malawi diet, which is 90% corn, 10% leaves. You see this radical shift in the microbiome. Give them the ready-to-use therapeutic food peanut butter-based supplement that's used in the clinic. They start to recover, switch back to the Malawi diet, and they crash again. And we can transplant these individual phenotypes from people into mice. So this is work that was led by Michelle Smith in Jeff's lab. So when you take a kid with kwashiorkor and give its microbiome to a mouse, the mouse does really badly. So it loses 30% of its body mass in 3 weeks and dies if it's untreated. But we can rescue it with the ready-to-use therapeutic food. And you see this profound change in the microbiome that we do not see when we see mice colonized with their healthy identical twins microbes, the healthy identical twin of the kid who has quash. So we see these individualized responses in the microbiome that are related to particular nutritional interventions and to the microbiome of the person they came from. So this ability to transplant from humans into mice really lets us use all the tools of systems biology that are available in the host to try to dissect the microbiome as well. And especially this technique of culture collections permits mechanistic hypothesis tests. So this was originally pioneered by Andy Goodman and then elaborated in this paper we did with Jeff's lab in Science 2013. But essentially what you can do is you can take hundreds of strains of microbes out of a single sample, grow them in pure culture under anaerobic or microaerophilic conditions, and then mix those culture collections together to prove that you can get a community that transmits a phenotypic effect. That's how you know for sure that it's a microbe that's doing it, and not, for example, a virus, not, for example, a cytokine or a small molecule metabolite, or any of the other things that you transmit when you transmit a fecal sample. So this really gives us the power to go in a lot of depth and figure out exactly who's doing what. So a nice way to think about this is if we're getting back to studying complex ecosystems on larger scales like Yellowstone, for example. It turns out that wolves have a tremendous impact on the Yellowstone ecosystem, but we didn't find that out by going there, grinding up a cubic mile of stuff and sequencing all the DNA, because if you do that, you're not going to find a lot of wolf DNA, and you're not going to realize that it's what's important in the ecosystem. The reason we know that is that people went in there, shot all the wolves, the ecosystem changed, then the wolves were reintroduced and the ecosystem changed again. And so the ability to do essentially this for the gut ecosystem and to go hunting for microbial wolves at the level of individual taxa is incredibly exciting and really the direction that all this is going to go now. So with that, I'd like to thank the large number of people currently or formerly in my lab. our large number of collaborators on various projects, and our different sources of funding, some of whom require I show you their logos when I talk about specific data sets. And finally, thanks for your attention. I'd be delighted to answer some questions. And I'm sure you have a lot of questions at this point. If we don't get to your questions, do feel free to email me after the webinar. Thank you.
Oh, thank you very much, Rob. This is fantastic. We actually do have a question, which we can address first off. And this came up when you were looking at the mouse model, the TLR5 mouse model. And the question is, does the age of different people in the same family affect the microbiome? For example, a child and the mother in the same family, are their microbiomes different? Probably came up.
Yeah, good question. So everyone's microbiome is different. I actually go into this in a fair amount of detail in my book that just came out on Tuesday called Follow Your Gut, The Enormous Impact of Tiny Microbes, where essentially everyone's microbiome is going to be different. The microbiomes of parents and children, if those children are older than about 3 years old, are more similar than the microbiomes of two unrelated people. And also you converge on your partner's microbiome as you live together. We don't know what the kinetics of that are, but people who have been living together for a year or more certainly have significantly more microbes in common, and we can even match you up to your dog by the microbes you share. Now, very young kids, like within those first 3 years of life, their microbiomes are so different that they're going to look more like other kids than they do like their parents. Although you can still see a signal of transmission between kids and parents.
Beautiful. We have some more questions coming in, and I'd like to invite you to continue to add questions, but I would like to bring Dr. Kuo and Dr. Young into the conversation. Would you each like to just say hello so people can hear your voices?
Hi, this is Howard Young. And Rob, what do you think about approaching disease when you don't have the facilities available like the germ-free animals? That you saw, the picture you showed is a very large one, but many places don't have that available. What is your sense of trying to understand the microbiome in conditions where you can't go to that system?
Yeah, good question. So without a germ-free facility, you're really limited in your ability to establish causality. You can use antibiotic knockdowns to colonize mice with mouse microbes, but if you use them, if you colonize them with human microbes, as soon as the mice are exposed to mouse microbes, the human microbes get displaced within a couple of days. And we have a paper in Cell with Jeff's group that came out last year that goes into that in a fair amount of detail. So when you co-house a mouse colonized with the human microbiome, when you co-house it with a mouse with mouse microbes, the human microbes just get displaced immediately. So you can do a fair amount with mouse models without a germ-free facility. You can look at the effects of adding a microbe without a germ-free facility if you can get it to establish. But then the drawback is to that is, first off, you might not be able to get it to establish in an already complicated ecosystem. Second, it's really hard to get IRB permission to give a microbe, especially a fecal microbe, to human subjects. So again, you wind up more limited to animal models.
Winston, would you like to say hello?
Sure. Hi, Rob. This is Winston. Thank you so much for such an outstanding and informative presentation. You showed in one of your slides in reference to data demonstrating vaginal birth, the cause and effect, and that our microbiome changes over time, as you see in kids. Is there a difference, because you mentioned most people are born through the vaginal way. Is there a difference between vaginal and C-section in terms of outcomes for the kids?
Oh yeah, absolutely. So I dropped that in the interest of time, but when you look at vaginal versus C-section kids, at least early on, and we did this with Maria Gloria Dominguez-Spallo, who's now at NYU. This was in a paper that came out in PNAS in 2011. Vaginally delivered kids are coated in vaginal microbes. C-section delivered kids basically pick up skin bacteria from their environment, not specifically from their mothers, and they look totally different, not just in the stool, but all over the bodies. Then in terms of health effects, C-section is associated with higher risks of asthma, of allergies, of atopic disease, and even of obesity. and all of those have now been linked to the microbiome. So we're starting to think that maybe the vaginal community is an adaptation. It always converges on the same state during pregnancy, right before delivery. So we think it might be an adaptation to coat the baby in the right kinds of microbes to allow its immune system to develop correctly.
Beautiful. We have another question I'd like to bring in and open it to any of you to address. And I have two questions from the same person, so I'll read them both. The first one is microbiome related to specific mental conditions, i.e. autism, excessive anxiety, and so on. And the second question is, in view of millions of possible microbes, if we engineer the microbiome, are we setting ourselves for bad surprises? We cannot predict the outcome.
So I'll answer the first one first. So for anxiety and depression, there is excellent mouse model work linking the microbiome to both anxiety and depression, and also in manipulating that state through probiotics. The human data that's available right now is a lot less compelling at this point, but a lot of people are interested in the topic, and there's a lot of studies in progress that I'm aware of. So I think we'll be seeing some very interesting things in that area. Could engineering the microbiome have unintended consequences? Well, sure it could. I mean, anything could have unintended consequences, and exactly the same sort of thing was raised about chemotherapy in the 19th century, when it was considered very exotic that you would take a chemical to modify your disease rather than doing bloodletting or other accepted therapies. So, while it's true that there are many chemicals that you would not exactly want to add into your body, that doesn't mean that very carefully chosen drugs are not enormously effective and really useful. So I think we need a lot of additional research to figure out which microbe is going to treat which condition. Could there be a microbial equivalent to thalidomide? We all hope that there won't be, but some caution is certainly required rather than just rushing ahead and taking any microbe you isolated from any environmental source and doing human trials with it. On the other hand, the FDA's official position on this is that most microbes would not fall under the GRAS guidelines, so you'd have to do an IND, and you'd have to characterize that microbial material very well before you were able to give it to human subjects.
So, Rob, along that line, let me ask about probiotics, since there's a major push in the everyday advertising about maintaining gut health by giving yourself probiotics. Is there any data to suggest that the gut biome can be actually manipulated by taking microorganisms?
Yeah, good question. So again, the animal model data is greatly in advance of the human data, and public enthusiasm for probiotics is greatly in advance for the actual scientific evidence. There have been some very nice randomized controlled trials at this point, with specific probiotics to specific treatments. So, for example, with Lactobacillus rhamnosus GG, there's been some studies looking at that and mitigating post-antibiotic diarrhea. Similarly with VSL3, which is a cocktail of a whole bunch of different probiotics, that's been shown to be effective for irritable bowel syndrome and post-antibiotic diarrhea, and I think one or two other things by this point. So there is some good RCT data out there, but the majority of things that you get at the supermarket, first off, there's no evidence whatsoever supporting the use of that particular organism for any indication, and second, the organism that's on the label is not necessarily the organism that's in the bottle. It turns out that it's really hard to grow things axenically, especially on a scale of thousands of liters, and what I think would be a fascinating project would just be to look at how frequently the label and the contents match for a lot of these things. Another thing that's an issue is that typically you don't get identification to strain level resolution on the labels of those products, but different strains of the same species can have totally different physiological effects. So it's a bit like you were getting a drug and it just told you it was an alcohol, but it didn't tell you whether it was ethanol, methanol, butanol, what, and you might care about those distinctions.
We have another interesting question that's just come in I'd like to share. And I will say that we are going to continue on for a few minutes. If you can stay with us, terrific. If you need to move on, thank you so much for being with us today. And this entire session will be recorded and will be on our website, and we will send a note out in a day or so. Here's the next question. Leaky gut syndrome was a hot topic at last fall's College of Gastroenterology annual meeting. Do you see anything in your data that indicates an effect of the microbiome on gut permeability? The follow-up question is, what does your research show about the relationship between the bacteria and fungal residence in the gut? I think that's two different questions, but maybe they're not.
Sure. Well, so for leaky gut, I personally haven't done a lot on that, although there's certainly a lot of interest, and especially the interaction between bacteria and different chemicals that we're exposed to in promoting leaky gut. So like for example, gluten intolerance, where there really is celiac disease, but a lot of people who are gluten-free don't have celiac disease. In the same way, there really is gut barrier dysfunction, but how leaky gut is colloquially used, I think, only maps onto that to some extent. There definitely is translocation of bacteria both into the mesenteric lymph nodes and into the bloodstream. So people who don't present with bacteremia in the sense that they're culture negative still have circulating bacterial DNA in their blood. And one thing that's been very interesting has been looking at the relationships between bacteria and atherosclerotic plaque versus what's elsewhere in the body, although intriguingly that seems to come more from the oral microbiota than from the gut. I guess that's a long way of saying that in mouse models, bacteria definitely affect leaky gut. In humans, there's a lot less data, at least that I'm aware of, but it's an active topic of interest, and so I'm sure we'll be seeing a lot more data coming out about it. In terms of the fungi, so only a couple of people have looked at fungi in the gut. Rick Bushman's lab at Penn has done some really nice work looking across multiple taxa. We recently, as part of the Sloan Built Environment Program, and in collaboration with Noah Ferrer at Boulder and Kabir P at Stanford, tweaked the ITS1 primers so that they work effectively on HiSeq and MiSeq, so working effectively on both platforms to read out fungal communities, but we haven't applied those on a large scale yet. It's the reason why you need to use different primers is that the 16S ribosomal RNA is different enough from the eukaryotic sequence that you won't get the eukaryotes when you amplify, and so you have to look at a different region. And for fungi, you really have to look at the internally transcribed spacer. So there's been some work relating the bacteria to microbial eukaryote pathogens, like for example Giardia, Blastocystis, and so on, but not very much at this stage linking it to the fungi. One thing to remember is that a lot of the stuff that I showed you would have been impossible 5 years ago and prohibitively expensive for most people 3 years ago, but it's very feasible now. So the vast majority of the studies that you would want to see done probably haven't been done yet, but are feasible right at the moment. And a lot of the reason why my lab spends so much time developing open technology platforms is that there are so many questions that you'd want to look at where the answers are going to be really interesting. But the best way to achieve that result is to enable a whole community of researchers to be able to use these tools to study all kinds of different things that we never would have thought of ourselves.
Terrific. Well, we have a couple more questions. Starts off with, it was a great talk, Rob. Thank you. This may be from someone you know, from Suman Roy. I hope I'm saying that well, at the National Cancer Institute. And these are two questions. What would be the future of microbiota in terms of therapy? And the second is, are we aiming for pills with specific microbes for different health conditions?
I'll answer the second one first. So specific microbes for different health conditions, definitely yes, just as in the same way that you want specific drugs for specific health conditions rather than just picking something from the pharmacy at random. The really interesting question at the moment is how much personalization is there going to be. So how much is what bacteria you take going to depend on you personally, as well as the disease you have, and especially some of the work trying to understand how bacteria can establish into a complex ecosystem is going to be really important in that respect. As far as the first question, which microbes are going to be used for therapy and how is it actually going to work? If I knew the answer to that, I'd probably start a company based on it, right? Like I don't think anyone really has a good handle on that, except for the case of C. diff, where Ceres Health has a product that's in phase 3 clinical trials. MRL and Virco's company, I think, also has a product in clinical trials somewhere, and so with that, it's a lot clearer because there are some clues like looking at the spore-forming components of the healthy gut that get you a long way towards being able to figure out what you should do. So. Let's see. I mean, there are some general, some of the organisms that are widely used as probiotics, like Lactobacillus and Bifidobacterium species. Many of those have very reasonable mechanisms of action, like producing butyrate in the gut, which is anti-inflammatory. But I think we're right at the stage of boiling willow bark or whatever, rather than having aspirin, let alone having COX-2 inhibitors. And I think there's a tremendous amount of research that's going to separate us from that. Although with the molecular tools we have now, I think it can happen pretty quickly.
I'm going to bring up one more question, and then we'll have a bit of discussion, and we'll adjourn in a few minutes. But this is an interesting one, and I think we kind of addressed this in the animation you showed in infants, but maybe you can address this in terms of adults. Do changes in the microbiome, gut or otherwise, revert back to a native state, or does the new microbiome thrive?
Great question. Both of those can happen. So there was a really elegant study by Les Defliths in Dave Rublin's lab where he gave 3 people antibiotics, all the same dose of Cipro, and basically one of them came back to more or less their original state in a few weeks, the second within a few months, and then the third was still pretty different at the end of the follow-up a year later. So, and then the sample size was 3, and they all responded differently. So all you can conclude from that is that it's going to be really different from different people, but we don't know how different. But that question about the resilience of the microbiome is really crucial. And there's very little data at the moment because historically time series have been so expensive to do. But it's really the key question, right? What happens after therapy, and how well do you go back to your original state? And is it even desirable to go back to your original state, or does that state predispose you to whatever condition it was that you had to get the treatment for? And we just don't know at this point. We're doing some interesting work with Janet Jansen on that topic in the context of inflammatory bowel disease, but the results aren't published yet.
Well, beautiful. We can take a few more minutes. Dr. Kuo, do you have anything that you'd like to ask or say before we part?
No, I just want to comment. It was a great presentation. A lot of detail, a lot of work over the past decade or so. No, thank you, Robert.
Great. And Dr. Young.
I'd just like to say thank you, Rob, for a great talk, and thank you for your efforts to make available to the research community the different tools, because I think that's critical, and I think anybody who's started to generate this type of data can quickly get overwhelmed by the complexity of it all. So your efforts are really going to help the community better understand the data they do generate.
Well, thank you. I would just like to remind everyone that this is the first session of a 5-session series of webinars. Next week, Dr. Kuo and Dr. Young will be, and Camelia Martin will join them in a discussion of a roundup of the news of interesting things that are going on right now in research related to the microbiome. So we'll be getting notices about that, but I do encourage you to go to our website and register, and feel free to send us any feedback. Many of the wonderful programs that we do were started because somebody said, I'm really interested in, or I would really like to present, or I would like to help you start something. And so I'm very, very much interested in hearing your thoughts. So I want to thank everyone for coming today. Thank you, Dr. Knight. Thank you, Dr. Young. Thank you, Dr. Kuo. And thank you to all of our attendees. Take care.
Thanks again.
Thank you.